Yes, this is metal carrier protein. Or yes, metallothionein and metal carrier protein. And that is the figure. And with the great charge of metal ions, this H group, cysteine group, promote great affinity to bind and to transport essential mineral ions as iron, zinc, copper, selenium, and in the microelements, and also cadmium, mercury, and lead. His great reducing capacity of the thiol group is H group of cysteine of metallothionein has important roles as a reducing agent inside the cell. As free radical scavenger, redox processes, and cellular redox condition, improvement of type 2 diabetes, improvement metabolic syndrome pathologies in clinical medicine and in toxicology. In the next figure, we see, we see the molecular structure of metallothionein. The molecular structure, you can do the next figure. Yes, we see the molecular structure of metallothionein established by Margosius and Vallée, Journal American Chemical Society, 1957, with a great number of reduction power, reduction power by its high number of cysteine amino acids. And in the next figure, also Margosius, in the next figure, Margosius and Vallée in 1957 established the mineral ion content after the discovery of metallothionein in horse cortex kidney. The percent concentration of metal ions per mole we see is cadmium, zinc, iron. In the next figure, please. The next figure. I cannot do the next. Yes. Thank you very much. We see the several mineral elements established by Margosius and Vallée. And the gradient affinity of some mineral ions are some as cadmium, zinc, copper, silver, bismuth, lead, and iron. In the next figure, we see that after Kage and Norberg, metallothionein, edited by Birkhauser, Ferrac, and Basel in 1979, metallothioneins are low molecular weight proteins of 7 kilodaltons, non-enzymatic activity, lack of aromatic amino acids, and histidine, and with 20 cysteines of 60 to 70, sometimes 70 amino acids. Invertebrates, metallothionein isoforms have about 70 amino acids. High percentage of thiol group, high metal content, 7 to 12 mineral ions, bioinorganic structure and kinetic lability, conferred catalytic activity by its ionic ligands, and great reduction power localized in all cellular systems, as in cytoplasm, mitochondrial, and for instance, also in nuclei of hepatocytes. Metallothionein is essential for the catalytic activity of enzymes and metabolism, because its prosthetic group or cofactor generally contain essential mineral ions or vitamins also, conferring its activity. In the next figure, we see the representation of molecular structure of mammalian metallothionein in its primary sequence, represented with zinc after Otvos and Armitage on the left, published in Proceeding Natural Academy of Science in 1980. And on the right, with cadmium ions by Radke Nedeki and co-workers in the International Journal Molecular Science in 2013, with 20 systems as crucial amino acid with ion H groups reduction, as a very reduction and active groups. In the next figure, we see the molecular structure of cysteine, glutathione, serine, and lysine, con similar amino acids, reducer amino acids, containing H groups, which are very reactive sulfur groups in its molecular structure. Cysteine are formed disulfide bridges on mineral element ions, binding transporting them to the systemic organs. Glutathione are both active redox agents to maintain the reduced environment of the cell. In the next figure, we see the important functions of metallothionein in metabolism and in toxicology. Free radical reducing protein, maintenance of the mitochondrial electrochemical potential, like glutathione, it maintains the redox state of the cell in cytoplasm, in mitochondria, etc., and other cell organelles, provides essential ions for the function of the electron transport chain, and regulation of enzymatic catalytic activity, implication in the homeostasis of the electrolyte balance. And in the next figure, on the graphic of metallothionein implication in some of them already mentioned function in toxicology, oxidative stress, free radical scavenger, wound improvement, enzymatic catalysis, etc. As we see in the next dia also. In the next figure, after Kressel and Maret, published in Biochemical Journal 2007, published the signification of zinc in metabolism as metallothionein-dependent zinc and redox regulation in two molecular forms, oxidized and reduced, as reductant and oxidant, in cellular signaling in multiple cellular pathways, in antioxidant defense. And in the next figure, Kressel, the same authors, Kressel and Maret, in the next figure, please, in Biochemical Journal 207, already mentioned, explained that the zinc availability binds thiol group or thiolic groups and oxidized metallothionein transporting them, the metal ions, to the systemic organs in redox-dependent processes of metallothionein. Thus, depending on the redox state of reactive oxygen, nitrogen, and sulfur species in the cells formed under oxidative stress, can also convert metallothionein, oxidative form, directly and release zinc ions. Releasing ions improves metabolism by the catalytic activity of enzymes, which prevent and benefit metabolic diseases, as shown in the next graphic. In the next graphic, Radke Nedeki from Poland and co-workers published in International Journal Molecular Science 2013, also related some metallothionein function on the cell growth of survival cell differentiation, cell resistance, differentiation, on cytokines, activity, free radical scavenger, etc., as before mentioned by other authors. In the next figure, we see very interesting in the role of metallothionein in obesity. In the next figure, please. The very interesting role of in obesity, the current global world pandemic. in the developed countries because regulate lipid metabolism and fatty acid liberation from tissues, which are released from the tissues in obesity people or in obesity subjects. In the next diet, the next dia, please. Yes, thank you. From tissues that accumulate in obesity the fatty acids. After Sato and co-workers in 2013, obesity and methylation in current pharmacy biotechnology published the regulation of lipid accumulation, lower than oxidative lesion, prevention the stress, prevention the stress of the endoplasmic reticulum, benefits the associated pathology and also of the metabolic syndrome. The authors mentioned that the pathology in liver adipocytes is improved by the metallothionein levels and regulation. The absence of metallothionein induced pathology, hyperplasia, excess of lipid accumulation, hypertrophy, and finally, obesity. After the mentioned authors. In the next figure, please, we see the metallothionein 2 in relation to physical activity and exercise. The stress, all diseases and the stress induce the high concentration of high levels of metallothionein. The exercise, the activity, and the stress benefits and enhances the function of complex 1 in the electron transport chain or phosphorylation in phosphorylation. Absorption and transport hydrogen ions and the electron mechanism of the source of vital energy and in physical activity. Protect to the complex of the electron transport chain for the toxic blocking and uncoupling effect rotenone. Rotenone is a specific inhibitor of complex 1 from the electron transport chain. From the Biochemical Society media publication 2006, it is in a manuscript. In the next figure, we see the electrons and protons, hydrogen ions, transport by the electron transport chain from NADH, nicotinamine, adenine, dinucleotide reduced, and to NAD plus free radical ions reduced by enzymatic activity, regulated by metallothionein, transported essential mineral ions. For vital energy production, for metabolism, intellectual activity, and physical activity, as we see in exercise. In the next figure, we see the energy synthesis as ATP, the energetic molecule for metabolism, intellectual activity, and physical activity. ATP will continue to form as the protons pass through the ATP synthase enzyme molecule, and it is forming the energy for our vital energy. In the next figure, please, we comment the Parkinson syndrome about commented by Ebadi and co-workers, published in Methods in Enzymology in 2005 about the neuroprotective role of metallothioneins. It is a neurodegenerative disease characterized by a progressive loss of dopaminergic neurons in the substantia nigra, zona compacta, and in other subcortical nuclei, with defect in the mitochondrial electron transport chain, as we see before. The lower oxidative phosphorylation and enhances oxidative stress with high concentration of free radicals, hydrogen peroxide, and hydroxyl radical hydroxyl. Low number of dopamine receptors, accumulation of neuronal toxins like 6-hydroxydopamine and methylphenyl tetrahydroxypyridine. And in the next figure, and we see that in figure, the immune response that also has to do with metallothionein, because essential mineral ions transported by metallothionein as zinc, copper, selenium, etc., induce to the immune system. Healthy young workers in China, mines in China, were expressed that they not has cancer, that people, the workers with high levels of metallothionein has no cancer in China, were published in several journals. And in nonagenarians with high levels of zinc metallothioneins have a good immune response. Interleukin-6 is pro-inflammatory, inhibits the release of zinc from zinc metallothionein and causes inflammation, induces inflammation of the interleukin-6, will reduce zinc availability. Zinc metallothionein is also a bioindicator of the operation of the biochemical cascade of inflammatory. Zinc metallothionein is involved in the development and maintenance of the immune response. And in the next figure, we see after Chida and co-workers published in Neuro in volume 7 in Cell Press in 1991, that published that metallothionein structure shows similar growth inhibitor factor structure from the human brain 7 and 20 kilodaltons, and deficient in Alzheimer's disease also, with analogous sequence and same structural properties. In the next figure, we see the comments of Mackay and co-workers in European Journal of Biochemistry in 1993, and also Bebiano in Portugal, Bebiano and co-workers. I know personally by congresses in Portugal in thousands and in analysis reported isometallothioneins in invertebrates, as with the same results as before in mussel, in methylocedulis, in the same kilodalton fraction, methylocedulis, 4 isoforms, 72 amino acids, 12 glycines in 5 species of invertebrates, with analog sequence in both cases. And also in 20 kilodalton fraction in methylocedulis, 5 isoform, 71 amino acids, 12 glycines, and also analog frequency in both cases. In the next figure, we see metallothionein isolation identification on the right. We express the usual rapid technique that we do in our laboratory: homogenization, centrifugation, heat precipitation, 95 degrees Celsius, 5 minutes, centrifugation, filtration, and HPLC, high performance liquid chromatography, or polyacrylamide gel electrophoresis, or capillary electrophoresis. And in the next figure, we see in our reports that in Advanced Physiological Science, volume 12, Nutrition, Digestion, Metabolism, 1981, from Pergamon Press, the isolation of zinc-cadmium metallothionein in fraction number 20 determined by atomic absorption spectrophotometry. You see that the concentration of zinc and cadmium, and the peak are both for zinc and cadmium radioactivity because the gel is cut in portions and measures the radioactivity. Appeared in similar concentration, both metallothioneins induced by zinc and cadmium. The metallothionein has a great activity for both metal ions. In the next figure, we see the isolation of metallothionein from mucosa of anemic rat. That's very interesting because it's transferred from mucosa to the liver, the iron in anemic rats, after administration of ferrous tartrate. Zinc metallothionein alone appears also in fraction 20 and also in 30 with iron, copper, determined by atomic absorption spectrophotometry. And two peaks of iron metallothionein. It is seen at the end, on the right. In the next figure, the separation of metallothionein isoforms by 10% polyacrylamide gel electrophoresis, all of them in the unique peak because it's very low percentage with no separation. In the next figure, now the separation in polyacrylamide gel electrophoresis of 15%, we see the separation of cadmium, radioactive cadmium 109 metallothionein from rat liver in polyacrylamide gel electrophoresis show 4 radioactive peaks, as we see in the figure. And in the next figure, now the separation 22.5% polyacrylamide gel electrophoresis. There are also seen more left, also 4 peaks of metallothionein. And in the next figure, with 33%, with the 33% polyacrylamide gel electrophoresis, are also shown the 4 in the left, 4 radioactive peaks of cadmium 109 isometallothioneins. And in the next figure, we see the separation of control rabbit kidney metallothionein and the peaks 2, 3, 4, and 6 by high-performance liquid chromatography, HPLC, in 1993 by Ribas and co-workers in trace element, analytical chemistry, medicine, and biology, volume 6. And in the next figure, the metallothionein is a form of rabbit kidney after administration of cadmium chloride by intraperitoneal route, separated by HPLC also, and they are seen 4 clear peaks of metallothionein. Cadmium has great affinity to metallothionein, as we see in the figure. And in the next figure, the metallothionein isoform or radiokinin after administration mercury. We see in the next figure the administration mercury, and also shown the metallothionein peaks. And in the 31 dias, the administration of lead, cadmium after cadmium mercury. Please, the next figure. The administration mercury, also by HPLC, it is clear how several peaks of metallothionein. And in the next figure is the administration lead, nitrate lead, also determined by high-performance liquid chromatography. Lead has not a great affinity to isometallothionein, and the separation is not so clear, but we see several peaks. And in the next figure, metallothionein isoform in rabbit kidney after administration of copper. In copper, we see also the great affinity of isometallothionein. Metallothionein has greater affinity than to lead, and we see a nice clear separation of the isoform. And in the next figure, metallothionein isoform in rabbit kidney after administration of zinc. After administration of zinc, zinc has a very great affinity. The next figure, please. That means we see the spectrum of HPLC, spectrum of metallothionein at administration of zinc chloride. Zinc chloride, 1 milligram per kilo rat rabbit was the great affinity of metallothionein for zinc-induced anion separation 2, 3, 4, and 6. Yeah. And in the next figure is the isoform metallothionein in beta cells of pancreas. Then in the next figure, we see the isoforms in beta cells, rat pancreas, metallothionein separated by HPLC also. And we also see the separation 2, 3, 4, and 6 isoforms of isometallothionein. It was in collaboration with a group in Barcelona, Spain. And in the next figure, the human liver metallothionein. Maybe the human, we have not great quantity of metallothionein, but very important, as we see before, because the workers in China with high levels have got cancer, and with lower levels has cancer. One peak is shown and need more studies, more experimental studies in human or systemic organs. In the next figure, by invertebrates, in fish, by trout, liver metallothionein, by capillary electrophoresis. It is shown also several peaks. And in lichen, in the next figure, in lichen was also studied in Usnea barbata, also sent by a group in Portugal. In Portugal, the professor Amelia Martins Lousao and also Cristina Branquinho from Lisbon was sent to Usnea barbata. We determined this and show by HPLC these peaks of metallothionein in several peaks who need experimental studies. And finally, the Diptera larva Parsarcophaga archaeolostoma, the common fly. The next dias, please. The common fly. No, the next one, please. In the before, in Parsarcophaga archaeolostoma, yes, we never, but there are not than the Parsarcophaga archaeolostoma, the fly cultivating in our laboratory. And finally, we send isometallothionein present in all species. We observe isometallothionein present in all animal and plant species have not been found in Anodonta. Anodonta has not been found. And in the next figure, as conclusions, this is the common fly, Parsarcophaga archaeolostoma, that we do in our laboratory for tests in toxicology. And in the next figure, the last figure as conclusions, we expose the importance of metallothionein in the physiology and biochemistry of the human body and of all living beings seem obvious. In the next figure, please. And its implications, in the second one, its implications in the benefit and improvement of the numerous pathologies, neurodegenerative disease, Alzheimer's, Parkinson's, type 2 diabetes, etc. The transport of essential mineral and ions for metabolism, enzymatic activity, production of intellectual and physical vital energy is manifest. Its signification in toxicology is also clear because it is induced by toxic organic, inorganic, physical, and psychological agents of stress by preventing its toxic effects, accumulated cadmium, mercury, and zinc, and prevent the toxic effects of metals in mammals, in humans and mammals. In toxicology, it is considered as an early bioindicator of exposure and toxicity involved in some molecular pathologies. And many thanks for your attention.